Good morning. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios. And today is the 7th day of November, 2021. I think that gives us the potential for 3 prime numbers. So that's got to be significant somewhere in numerology, right? So what I want to do is get started with this presentation, and I'm going to do it right now. Okay. So I'm talking about the dialectical event ontology of immunosenescence as a harbinger to aging and ultimately the sickness unto death. And this is part 2 of the ending of the series. So we've talked about in the past this process known as aging or illness-associated immunosenescence. And basically it's a phenotype of an immune compromise. So it's not so much an immune deficiency or of a immune overreaching, but it can go to those extremes. So we'll call it compromise. We'll compromise to call it that, right? And basically what it involves is some kind of inappropriate targeting of host cells. So in a way, it's also an autoimmune disorder. That's what immunosenescence is. So the phenotype is first characterized, and because of that, has been classically described as an immune deficiency. But I think that fails to describe the real molecular subtlety of what controls the responses and how you make a transition from, say, poor surveillance and attack of true pathogens to some kind of misguided overcompensation, a frank overcompensation. And that can occur with commensal organisms. This happens as we age. And this, again, is giving you the molecular, drawn-out definition of immunosenescence. What it basically results in is a pathological reorganization of what would be normally some kind of protective system. Now, if you're talking about mucous membranes, such as the small intestine or the oral cavity, which are open directly to the environment, what you do is you lose the biofilm. And the biofilm integrity then relates to the pathology of immunosenescence. Now, that's only one region, but it's something to consider when we're talking about invading pathogens, for example. So immunosenescence can include being inefficient in mobilizing a defense using memory cells, particularly T lymphocyte and B lymphocyte memory. And you know that this is something that's in the news these days in terms of vaccinations, right? So basically what else it involves, immunosenescence, is a decline in a certain specificity, and there is some specificity, of the expression and differentiation of activity of natural killer cells, which come through the CD8 positive lymphocytic lineage, as you might recall. Now, this is going to involve a lot of epigenetics. And what I want to do this morning is bring into full on the floor so that we can examine it closely, the epigenetic component of the aging human phenotype and how that relates then to the morbidity and mortality, which we're trying to reach some kind of dialectical event ontology description thereof. So what else happens in immunosenescence besides this overreaching is you also get an inefficiency in detecting and eliminating tumors before they become metastatic. And that's because there's a lack of a phenotypic discrimination of tumor cells from non-tumor cells. Of course, that's going to relate to lymphocytic immune activity. We've been talking about this a great deal, so it shouldn't be a surprise to you. So you have aging-associated malfunctions, and they appear and sometimes present even in younger individuals, even people in their 20s and 30s. And this has been well described in the obese population. the obesity pandemic, if anything, has gotten larger or more gravid since the last 2 years. And this obesity epidemic is linked to an increase in the rate of acquiring an immunosenescent phenotype. So younger and younger people have what looks like increasingly an aging immune senescence. So this is something that's of significance, not just when you think about invading pathogens, but you think about the overall deterioration of the human condition. So in a way, obesity is a progerian, non-stable, prodromal state for immune compromise. Excuse me. So that's first thing. Okay. Associated with this can be metabolic, psychiatric, and cardiovascular disease pathologies. And you also get the strong representation with cancer patients. And this can be because the cancer patients may also be suffering from chronic autoimmune states for reasons we've gotten into already in the lectures. So immunosenescence is not due to a lack of immune cells, but it's due to a reduced repertoire of diversity of immune cells. And then because of that, an insufficient production of the correct lineage of immune cells. So this all means that you have a diminishment of naive T lymphocytes. Excuse me once again. I've got something in my throat. I think I'll take a drink. So the immune compromise doesn't have to do with the lack of a sturdy immune cellular lineage in circulation coming in and out of the lymphatic system. No, it's not the case at all. It has to do with the immune cells that are available and whether or not they're functioning correctly, meaning they're receiving the correct signals and they're responding to those signals appropriately, but also whether or not they can differentiate into specific T cell, and we're only talking about T cells here. We can talk about other cells, and we will later, lineages that will respond more specifically to the vagaries of whatever compromise or stress phenomenon is occurring. And again, this is all increasing as we age. Okay, so immunosenescence can be linked to what we've been talking about, describing an aging-linked, I guess, an atrophy, but more specifically anatomically, pathoanatomically, a thymic involution. And that phenomenon has been well described for many years. That's why we have a name for it in all kinds of vertebrates, including, of course, the models we use in medicine, mice and rat models in particular. And basically, it's a functional decline process because when you get thymic involution, or that is atrophy of the thymus as you age, you lack the ability to generate new T lymphocytes that are of the naive to secondary stage of differentiation. And so what you do is you corrupt T cell homeostasis. Okay? And so once that occurs, once you get a corruption of T cell homeostasis, you're on the way to getting an immunosenescent phenotype, which again is normally linked to aging. But more importantly, that what we mean by immunosenescence is this lack of control of the immune response. And this occurs sometimes pathologically because of other disorders that can be brought on by a more global disoriented metabolic activity. And this is often linked to negative association with poor nutrition. And sometimes in years gone by, some of that could be because of essentials in the diet being deficient. certainly was the case in prehistoric and historic times, probably all the way leading to the invention of modern medicines that's talking about it being the late 20th or the late 19th through the 20th century. But more importantly, this is all associated now, not so much with the lack of essentials in the diet, but the lack of absorption and then homeostasis from nutrition, leading to metabolic homeostasis and cellular mediated health. And all of that becomes corrupted because of obesity. And this is now the new problem, right? So let me hide this bar here. I didn't realize it was on. Let's move on. So you've been listening to me. I'm Dan Guerra, Authentic Biochemistry. And let's move on from that discussion to our next slide. Now, there's a lot here in this slide, so I'm going to go through it, pull myself kind of off stage here. All right. So it's a paper you can see that was published a little over a year ago. And what it tells us is the following. It's very important to get deeply involved in the understanding of the biology as well as the biochemistry. Aging, of course, is both necessary and it's universal in humans. So it is indeed an aspect of an a priori synthesis. You see this in particular what we're describing, heterotrophic multicellular organisms like humans. And it's particularly because we reproduce by sexual reproduction. We cannot survive simply by somatic division. That's a key feature of the aging process. And I've talked about this in the past because I talked about the evolution of sex in biological systems a couple years ago. I'm not going to rehearse that and redo it right here, but you realize that even sexual reproduction is something that had to appear in the biota for us to be able to start examining it and understanding it. And where sexual reproduction allows for a zygote generated from that fusion of egg and sperm to bring on a sort of hybrid vigor because of two different genomes fusing, two different haploid genomes fusing in the vertebrate animals like in humans. You also get with that a lack of being able to reproduce clearly just by cell division. And cell division just means replicating the genome one copy at a time, and of course acquiring mutations along the way. It also means a faster growth rate. But it does add a sort of immortality to the system when you think about an individual cell, bacterial cell, let's say, dividing and dividing and dividing. The original cell, that the cells that exist today are derived from, could have been quite ancient, you see. Certainly not following the aging processes we do as heterotrophic as well as sexually reproducing organisms. So no simple mitotic division. And essentially what happens in very significant association with that is we get terminally differentiated cells and tissues and organs. And so the only time that can be redirected in terms of cellular differentiation is during tissue repair and, of course, unfortunately, oncogenesis. This is after the developmental sequence. So you get a unidirectional sequence of lifespan, and it's measured as cellular and physical biochemical systems. And that involves then a senescence. And the senescence has to do with the many mechanisms we've already talked about. These, of course, include telomere shortening, DNA mutation, and epigenetic modification, all of which are related to the immune mechanisms, which can tailor, modify, and promote or suppress those major mechanisms. All those mechanisms, though, leading to an increase in cellular aging, and that senescent process itself ultimately leading to multiple morbidities, because we're multicellular, you get multiple morbidities, and then all that linked then to the ultimate processing of life called death. So, as I write here, you get a temporal alteration of transcription factors and protein-based signaling trafficking patterns, membrane topodynamics, that is surface-to-surface dynamics, including, for example, rafts, right? Membrane rafts, moving back and forth receptors to the surface of the plasma membrane, thus altering the signaling phenomena. And then subsequent to all that signaling at the surface of the cell, everything distal to that response, meaning intracellular, including alterations in gene expression. So that necessarily involves a bioenergetic system, that is the production of ATP, and all of those phenomenon are episodic, even though there is a final terminal cell fate, which is going to be either senescence, where it no longer replicates, or necrosis, where the cell actually degenerates, dies, or oncogenesis, where the cell inappropriately goes through multiple divisions, and then in so doing, usually because it's been removed from the temperate control of cellular division, which means it becomes tumorigenic. Or, of course, the cell can reach the fate of programmed cell death, such as apoptosis. We've talked a lot about the subdivisions of each of those in the past. So you get physiological aging, which results from an incremental organ damage and ultimately failures. And that includes then an increased susceptibility, because all the organs in the body are involved in the immune response, as we've been talking about. You get increased susceptibility to diseases because of etiologic agents, and of course the environmental stress, such as xenobiotics, and of course a stochastic, meaning random plenum of mutation, epimutation, and oxidative degradation because of the presence of molecular oxygen having unpaired electrons. And collectively, we understand that as a pathobiochemical morbidity, which is what we observe in all humans, but particularly increasing as we age. So aging, and especially unhealthy temporal decline, and that temporal decline is euphemistic for aging, right? Associated obesity are primary risk factors in the major human diseases that ultimately progress towards death. Cardiovascular disease and cancer and autoimmune disease are three that often come up, right? So major human diseases that chronically accelerate during aging toward death include, as I just said, CVD, various forms of cancer, neurodegeneration such as AD and PD, and of course the metabolic disorders. With one that comes to mind, type 2 diabetes, for example, or just metabolic syndrome, associated, for example, with high blood pressure. So there has been an increasing effort to uncover the role of epigenetic mechanisms in both chronological aging and in this subset of processes called cellular senescence, or aged cellular mass, teleological movement towards the end, right? Now, these epigenetic changes are rendered via environmental stress. So that includes all the internal possibilities, such as the production of reactive oxygen, right? And all the external vagaries, such as pathogens, but also xenobiotics and any kind of tissue damage, including things like microstrokes, like TIAs, right, which happen all the time. And I've described a lot of these in lectures, so I hope you remember them. We talked a lot about these in the audio lectures on the podcast. But remember, this involves lipid fluidity. It's very important. That includes the understanding of what the incorporation of polyunsaturated fatty acids do to membrane dynamics related to the movement of proteins, for example, signaling proteins or receptor proteins or channel proteins, thus the entire associated comprehensive communication network across the plasma membrane. All of that is linked to membrane fluidity, which can be directly biochemically associated with polyunsaturated fatty acids, and then the lipids in which those fatty acids are bound, usually as oxygen esters and sometimes also associated as sulfur thioesters. And of course, we have to keep in mind that there are also ethers, such as in the plasmalogens, right? We've talked about these. So you have ester ether linkages primarily here with fatty acids. Those fatty acids with multiple double bonds are going to be prone to autooxidation because they have unpaired electrons, but also they're going to be subjected to oxygenation. And remember those discussions of the cyclooxygenases, lipoxygenases, epoxygenases, and P450 monooxygenases, all those enzymes then that are going to relate to the incorporation of oxygen into those double bonds. Okay. So, epigenetic changes that are rendered then with environmental stress are very important. And when we think about the epigenetic changes, one of them is lipid fluidity. And I think I will make a good argument. I've been making a good argument for this. Because epigenetic doesn't just mean at the level of nucleic acid, right? Yeah, most of the time we talk about nucleic acids, and here we speak of, say, methylation acetylation of histones, DNA methylation acetylation, and then, of course, those small non-coding RNAs or interfering RNAs. But even indeed, epigenetic modification can be found within the dynamic event of chromatin remodeling, nucleosomal skipping and positioning during gene expression. And then you have to engender then also the tissue-specific gene expression patterning that will work through plastic and elastic molecular mechanisms, sometimes due to differential expression because of promoter activation or enhancer deactivation or suppression of both therein, but also because of the epigenetic modifications, slowing down or enhancing gene expression because of something like methylation slowing down and acetylation increasing because of alterations in heterochromatin to euchromatin, a discussion we've had along this series of lectures for well over a couple of years. So you have protein and DNA methylation, and those are common covalent epigenetic biochemical processes. You also get membrane lipid methylation and acetylation. So we must include those. We have the histone, this is the canonical histone lysine methylation patterns and histone lysine acetylation patterns. We have arginine methylation, not just lysine. And we also have lysine, as I said, methylation, acetylation, and indeed serine phosphorylation. Now, there's more covalent modification I'm describing there. Those are just the canonical ones. All those modifications alter the extent, this is again an understanding of epigenetics. The modifications I'm talking about, those covalent modifications, alter the extent to which DNA is wrapped around its histones. Okay? Now we're talking about nucleosome arrangement in chromatin. With that, the availability of a specific gene within that DNA, the open reading frame, to be recombined, replicated, repaired, and of course, because we're talking about gene expression, transcribed. So there could be a unique role for DNA methylation in aging. And today we're going to cover some of that, but we're also going to do a fair amount of induction in the lecture. That was a long introduction, but you didn't have to see me reading it the whole time, right? So that must be better than what you sometimes have to endure. All right, so let's move this again out of the way so you can see I'm still here. So again, more details about this epigenetic phenomenon. DNA methylation modification involves two different processes: the addition and removal of a methyl group at the fifth position of a cytosine residue, or indeed even the sixth position of an adenine residue in DNA. So you have DNA 5-methylcytosine, 5-MC. Clearly, that's the most prevalent kind of epigenetic DNA modification. And in fact, the most common DNA methylation. And that you see throughout eukaryotic organisms. Primarily occurs on cytosine residues. It's a pyrimidine nucleotide. And it precedes, the cytosine will precede a guanine nucleotide. So that's what we talk about. C, P, that's the phosphate ester bond. G, sites or islands. The more CPG iterative islands you have, the more common those are hotspots for this potential, and it's still only a potential, DNA methylation, generating as a product 5-methylcytosine. So when you have the presence of 5-methylcytosine, it's generally believed to prevent or to suppress transcription. And that's because transcription factors can't bind as effectively to the promoter region because you have this methyl, this kind of bulky methyl group right there where you want to have the machinery, for example, for transcription, the RNA polymerase II and the single-stranded binding proteins, and even the opening up of the DNA to get to transcription or, of course, for repair, but the gyrases and topoisomerases, right? Again, the single-stranded binding proteins. All of those proteins can be hindered because you have this hydrophobic methyl group stuck there, where many of those enzymes require a higher aqueous microenvironment. But a methyl group there, you kind of remove the hydrophilicity of that domain, of that molecular synaptic, if you want to call it that, a transcriptional synapse, I guess we could call it that. That domain becomes less hydrophilic when you start adding methyl groups. And it's not just one methyl group, of course, because there's going to be multiple CPG islands. And if it's on a promoter region of the DNA, so that I just described as an organic chemical stressor on the opening and availability of the DNA for transcription. But also keep in mind that the protein may not be able to process a methylated nucleotide because it's looking for specific nucleotide sequences, because that could be in a promoter region, and that's where you tend to have hotspots of CPG islands. So see, it's a normal regulatory mechanism for all eukaryotic gene expression, and it's we call it now epigenetics. So who knew when genetics were first being discovered in bacteria, plain old E. coli back in the late 1950s, Jacob and Monod, for example, and Shango, we shouldn't forget him, doing a great deal of work on our understanding of an operon and promoter regions and enhancer regions and how you get RNA polymerase binding. Of course, in the prokaryotic sense, much more linear, smoother level of transcription directly to translation, right? In the eukaryotes, we found much more complication. Well, one of the complications we were not aware of is this epigenetic programming, right? Sensing a CPG island, writing a methylation pattern, reading the methylation pattern with like bromodomains, and then erasing those methylation patterns, only to come back again, but maybe not in a specific manner, but in more of a stochastic or pseudostochastic manner, thus regulating gene expression. And now we know this is extremely common. Right? So I'm telling you that this process, because it's very well described now, and it's understood, we understand what enzymes are involved, we understand what the products and substrates are, but we don't yet know why there appears to be a fair amount of stochasticity, that is randomness associated with this. Except it looks like that randomness is what is required to alter gene expression in the individual genome, either at the cellular, tissue, or whole organism level, thus finally describing that agentic, individual, dialectical event ontology of biology for each person, because each person is going to have a different epigenome as a result of all of these changes in epigenetic signature modifications, right? And that's why identical twins, even with this basically the same genome set are going to develop and will live lives uniquely, even if they have phenotypic features which are quite closely aligned, as we know with identical twins, right? All right. So the presence of 5-methyl-C is generally believed to prevent transcription factors. Okay, so that's a good way to keep it in mind. And it prevents the promoter region from getting bound to all of those transcriptional elements that are necessary. So basically, you get with 5-methyl-C, at first blush of understanding, is a suppression of gene expression. Now, 6-methyladenine, okay, 6-MA, now we're talking about a purine methylation, is more recently described. It is indeed an epigenetic modification. It's an enzyme that only selectively methylates adenosine. And we do find these in the human genome. And so these are part of CpG islands. This is an A, right? And we actually have found, studies have described that this 6-MA signature, which goes through the same process of sequence familiarity to the enzymes responsible for changing them to a writing of a epigenetic signature, and then a reading of the epigenetic signature, like with the bromodomains, and then ultimately the erasure by things like DNA methylases, right? And all of that seems to be linked to mammalian development. And this has been determined recently in the last 10, 20 years. So let's take a look at this process in more detail. Let's get me out of the way again so I can carefully describe this to you. So you're looking at chromatin. You look at chromatin unraveling into nucleosomes. That's what these patches are. And then when we get to finer and finer magnification, you see double-stranded DNA. Right? I guess I could put myself in here a little more so you can see me looking at it. Why it would matter, I don't know. And so you see these CpG islands, right? You have complementarity. G binds to C, C binds to G. So this would be a CpG island because this is a double-stranded DNA, don't you know? And the individual cytosine residue has this particular pyrimidine ring structure. And so this is the methyl group we're adding to that C5 moiety, right? Now, you can also have adenosines methylated. We just talked about those. And you can see there's there's a nitrogen atom, and you're methylating that nitrogen atom, right? So that's what we mean by 6-MA. This is what we mean by 5-C, 5-MC, right? So this is 5-MC, and this is 6-MA. Okay, so now you know what they look like in some detail. All right. So you have a writer, and this would be something like N-sex adenosine methyltransferase 1, finding that adenosine residue there, the double-stranded DNA. And then you have erasers, such as this ALK BH1. So you can remove it, you can write it, you can remove it, but also it has to be read. And it's read again by these MBD proteins, some of which are known. Another name for that protein is one that had those domains are known as bromodomains. And I'm not going to go into the whole prehistory of how we call them bromodomains. It has nothing to do with bromide. Okay, it has to do with the Latin use of the word for bromodomain. I'm not going to get into the detail of it now. But this was all described in Drosophila genetics. That's all I got to say. So give it to those folks, right? Modulation of DNA methylation in mammals, DNA 5-MC methylated sites, the fifth position of cytosine, while 6-MA, of course, blah, 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 6 of adenine. The methylation marks are established by writers. Those are the methyltransferases I just described to you. Modifications are identified by readers. These are the ones that have the methyl CpG binding domain proteins. And then you have the erasures. And these are two different kinds of proteins, the TETs and the ALK BH1s. So those are two different enzymes which will remove that covalent methyl group, either from a cytosine or from an adenosine. All right, so enough of that. So what are some of the mechanisms then? DNA methylation, you have 5-MC, 6-MA. and they are going to require unique enzymology. So let's take a look at the proteins. So the 5-MC methylation is a result of a DNA methyltransferase, and there are several of them that have been described just by pulling out the sequences and finding different genes for them in the human genome, the Genome Project. But more than that, there's only 3 of them that seem to be active and related to functional alterations of gene expression. That's DMMT1, 3A, and 3B. Okay, so those are different proteins, right? So DMT1 is primarily responsible for the maintenance of methylation status. It's called maintenance methylation, and that can happen even during and after. So maintaining that methylation pattern as cells go through division, while the 3A and 3B work in a de novo mode. So rather than maintaining the methylation pattern to suppress certain regions of chromatin, and that's how that methyltransferase 1 functions. The methyltransferases 3A and 3B are brought in during the various stages of cellular life, and they find specific cytosine residues or adenosine residues, and then add the methyl mark on those. Now, what induces those enzymes can include things like stress phenomena, but also, of course, development differentiation, and then whenever stress has been recognized, repair processes directly into the cells that have been modified or stressed, but also repair and modification of entire tissues. And this again involves the immune cell lineage. In fact, the immune cells themselves will be heavily altered for gene expression via methylation and indeed acetylation, et cetera, those covalent modifications. So you have to think about the immune cells are prone to this covalent modification. We call it epigenetic, which alters gene expression in those, say, T lymphocytes, B lymphocytes, neutrophils, basophils, macrophages, type 1 and 2. We've talked a lot about this. Well, all of those cells can go through epigenetic retailoring. And when that occurs, of course, you're going to have an alteration of the function of those lymphocytes and leukocytes. So 5-methylcytosine methylation can also be oxidized, of course, and changes into, you get a 5-methylcytosine converted to a 5-hydroxymethylcytosine, so a 5-HMC. Okay, and it's done so by an enzyme known as 1011 translocation enzyme. And I've described that in detail before too, why it's called the 1011, and I'm probably not going to do it this morning because we're trying to talk about aging, not about these really interesting enzymes and where they have unique names. But let's leave it for now to say that there are 3 TETs that are functional here that will carry out this hydroxymethylcytosine production, and they're called TETs 1 through 3. Okay. Here you go, sort of the pathway laid out for the different methylation patterns, including that hydroxylation I just introduced. So we're going to call these pathways of cytosine demethylation mediated by 1011 translocation proteins, TET proteins. And the AID, which is the adenosine deaminase, and the BER, which is more of a process, not just an enzyme. That's base excision repair. We talked about this when we talked about DNA repair. This was back in the early spring, late winter of 2021. DNMTs, of course, are DNA methyltransferases. 5FC is 5-formylcytosine. Yes, yet another covalently modified cytosine. The 5-HMC we already described, it's hydroxymethyl. And you also have hydroxymethyl-uracil, and of course 5-methylcytosine. And you have TDG, which is the enzyme responsible for some of this final end stage modification, and that's the thymine DNA glycosylase. So you see, it's not just a few enzymes, there are several enzymes. And yes, indeed, there are different isoforms of them with differential sequence-specific substrate localization and activity. Okay. So now, if that's not enough, you of course also get the processing of removal of a hydroxymethyl group, which is right here, hydroxymethyl group, okay, by passive diffusion. And passive diffusion means a DNA replication. So you can remove that 5-hydroxymethyl group just when you replicate. So the DNA polymerase removes what it sees as dysfunctional or inappropriate nucleotide, in this case, it's inappropriate pyrimidine, and repairs it. Right? So that's recombination repair, replication repair, right? And there's a surveillance that goes on with DNA polymerases we've talked about before. It's a great processive enzyme. So we have passive dilution, removing that because when we replicate that genome, we don't replicate it as a 5-methyl hydroxy, we replicate it as just cytosine. Okay, that's how you get rid of it. All right, but here's the regular canonical process, if you want to call it that. Certainly it is in epigenetics. There's the DNA methyltransferase making the 5-methyl right there. There's the TET enzyme adding that's the 1011, right? Adding that now oxidation, which gives you an OH group on that. That's hydroxymethyl. More TET processing is going to give you more extensive oxidation of this. You're going to have now the 4-mil. You're going to have 4-mil cytosine. And finally, 5-CA cytosine. Right? So all of these different groups are potentially available as the carboxyl cytosine by the complete oxidation of the methyl group, right? So you go from a methyl group reduced to an alcohol, right? And then you've got the next phase, the 4-mil group, which is basically the same oxidation state as an aldehyde. And then you've got finally the carboxylic acid, right? So different stages of oxidation, all on that one cytosine residue. So 5-methylcytosine doesn't have a great deal of metabolic activity except this possible passive dilution effect. But all these oxidized forms do, the hydroxy, the 4-mil, and the carboxycytosines, all can be reacted with enzymes. This AID was one of them. This is the adenosine deaminase. And then following that reaction, you made a uracil now, a modified uracil, which you can only get an RNA, right? But you can get then TDG, right? This is the glycosylase, the thymine DNA glycosylase, and then go through base excision repair, DNA repair, and you get back to a fully functional, non-covalently modified cytosine. And it's going to occur again with the other further oxidized forms, the 4-mil and the carboxy. So you see there's a great deal more epigenetic biochemistry than we normally talk about. Great deal more. Now, what these different modifications do when they occur in situ without a faithful round of DNA replication, but let's say directly to a transcription or directly to a recombination, right? For example, with making a T cell receptor or the VDJ recombination for the B cell immunoglobulin series. These partially oxidized forms of cytosine epigenetic modification production will give you a slightly different alteration of ultimately the expression of the genes that had those signatures. Right? And that, of course, is going to modify things like cytokine, chemokine, growth factor, and indeed even, transcription factor expression in lymphocytes. Okay. And now finding all of those micro epigenetic derivatives is not a trivial task. It's very complicated in terms of laboratory research. And to be able to isolate those various intermediates, or they're basically the products of those reactions, and then determine where they are in the DNA. So you have to do a sequence analysis, and then you have to determine whether or not it's in all cells or only in some cells. And then finally, did you get a functional alteration? And the readout for that would, of course, be gene expression. Again, doing that in a snapshot way is easy, but doing it in a dynamic disequilibrium context, which is what living systems are, dynamic disequilibria, therefore they are events, right? That's much more difficult to find that kind of precision even in the laboratory of 2021. So we don't know much about it. We know it occurs, and we know it when we see it. And when we see it, we know what's going on. We know the chemistry and the biochemistry, but we don't necessarily know the physiology or the pathophysiology associated with it, at least not at the level of always being sure, because we can't always measure these things. Basically, that's what I'm telling you. So the methyltransferases can also act in reverse, as you might guess, and they're responsible for DNA demethylation. 6-methyladenosine methylation is generated by this enzyme NGAMT1 and by this ALK-BH1. But the level of 6-methyladenine in cells has not been consistently confirmed in specific, let's say, promoter regions of genes. Where it's been discovered once or twice, you go back and look, you don't see it. But of course, it could have been easily erased by them. So we don't know if this is a very, very random event or if it's as common as 5MC, generation, reading, removal. We just don't know yet. But we are starting to put together a library of 6-methyladenine signatures in DNA, and that's forthcoming. But as I said, it's very difficult to get all that work done at the same time trying to associate a function with it, right? Okay. So sorry for all the extra reading again. So I'm going to pull myself out so you don't see me standing there, sitting there, I guess, reading. It's a paper that was published a few years back, but has tremendous significance to our topic today. Published in Cancer Science in June of 2014. So yeah, 7 years ago or so. This is going to tell us a little bit about the TET family genes. So the 1011 translocation family genes were initially found as a chromosomal translocation partner in leukemia patients. And it turned out to be, after studying the enzyme, okay, to be really significant in DNA methylation. Yeah. All right. So this is the key feature of biochemistry. You first need to know what the healthy biochemical processes are. And then when you start finding alterations in what you think are standard operating procedures in DNA biochemistry, the next step is if we find changes in that. And that's how the epigenetic flow of information comes. First, you have to get an understanding of what is normal methylation pattern, a methylome, which is only a part of the epigenome. And you study that by epigenetic modification, you see. And then you have to then look for, if there are any, pathogenic markers. And then any kind of distancing or change in valence from the healthy to what we're calling now the pathogenic or pathological methylation pattern, if that is indeed associated with specific functional alteration, or is it just going along for the ride? It's another entire aspect of determining any of this epigenetic signaturing and reading and then ultimately removal, whether or not that has the primary effect on pathophysiology, or maybe even in superphysiology, such as acquiring characteristics such as in the learning process and the central nervous system. And here we're talking about neuronal epigenetic modifications, which can also go awry, as you might guess, to neurodegeneration, and that includes Alzheimer's disease and Parkinson's disease, prefrontal and age-associated dementias, just to name a few. Oh, and the other, of course, would be, brain cancer. So I heard back these TET proteins, first described as chromosomal translocation partners in leukemia. You know, leukemia diseases are diseases of aberrant chromosomal translocations. That's how they were first discovered. Philadelphia chromosome was one of the first ones discovered. So these TETs, as I showed you in the last slide, hydroxylate 5-methylcytosines, and then they can become converted to the 4-mil or carboxy, and then all the way back to an unmodified cytosine. So this parading through the oxidation state of that added one, C1, that carbon 1 on that cytosine residue, right, may be a way to remove the methyl signature, but it takes longer. Now, that slower processing, the oxidation of a carbon, ultimately we're getting to its removal. We've seen that all through living systems. That basically is what? Bioenergetics, right? That is eukaryotic aerobic bioenergetics, taking a reduced carbon and oxidizing it, in those cases, ultimately to carbon dioxide, and deriving energy from that process, right? So here's yet another biochemical motif used for something quite different. Here, maybe modifying that methyl group to eliminate it as a signature, but we wonder too, because there are multiple steps here, and there therefore could be confusion and error in those steps, if any of that has anything to do with the plenum of morbidities we see associated with aging, senescence, and then ultimately the high morbidities leading to mortality. Things we still need to study. So, somatic mutations of the TET2 gene were reported in a variety of human hematological malignancies, as I told you. So it's first picked up in leukemia, and then what's known as the myelodysplastic syndrome, which is basically prodromal to leukemias and to some extent lymphomas. And then leading to malignant lymphoma. So it suggests that TET2 is involved in the whole process of hematopoiesis, coming from those primary myeloid lineages or lymphoid lineages from the bone, but hematopoiesis. So the TET-mediated cytosine demethylation pathway is linked to a mutation of, this is where you're listening to authentic biochemistry, a mutation of isocitrate dehydrogenase 1 and IDH2, isocitrate dehydrogenase enzymes. We've talked a lot about these, either in the mitochondria or in the cytoplasm, haven't we? Yes, we have, in terms of regulating patterns of acetylation and deacetylation with the HAT-HDAC system. Remember all of that in various disease states. Once again, now we're looking at isocitrate dehydrogenase, which normally would just be what? It would just be an enzyme in the TCA cycle, generating NADH, of course. Okay, so it does something totally different here. So TET-mediated cytosine demethylation is linked to a mutation in IDH, and we find these in these hematological malignancies, such as the myelodysplastic syndrome and also leukemias and lymphomas. And the mutation that we're talking about inhibits TET function. And it does through a very novel metabolite called 2-hydroxyglutarate, or 2-HG. And we're calling that now an oncometabolite because it's associated with tumorigenesis. So mutant TET2, which again were linked to these blood-borne cancers, right, results in the expansion of a multipotent as well as myelogenic progenitor line. Multipotent meaning that they're back to mesenchymal stage of differentiation. And that can lead to an accumulation of multiple pre-malignant clones. Not truly malignant, so they start dividing without control mechanisms, right? So, in addition to the cytosine demethylation we just talked about, right, getting you back to where we were 5 minutes ago, TET are also involved in chromatin modifications and other cellular processes through the interaction of oxygen-linked beta-N-acetylglucosamine transferase. All right, now we've recognized this enzyme before when we talk about lipid metabolism. So now we're bringing in lipid metabolism into our discussion of immune systems associated with DNA repair, DNA epigenetics, all of this linked either to a frank disease state, such as the hemopoietic cancers or prodromal to those myelodysplasias, and ultimately to senescent processing and epigenetic signatures that are or are not maintained because of this oxidation state of the methyl group. Right. So you see how these things fuse together and come together, and imagine all these mechanisms working in a very finely, highly oiled, cool processing throughout many, many, many years in many cells and tissues, even after alterations of gene expression for various other alternative pathways. Ultimately, any potential mutations or epigenetic modifications could lead from a subtle all the way to a very profound change in the health of the cell and therefore the health of ultimately the tissue, the organ, or the system, leading to morbidities and mortalities, all of which linked with aging. So fully functional TET2 is actually a critical regulator of hematopoietic stem cell homeostasis. Now, that's a very profound discovery. And that was first made, or first really described in detail in this Cancer Science paper 7 years ago. So, once again, you get the idea of how things are always changing in literature. And that's why it's absolutely necessary to be reading the current literature, right? The literature that's been published even a year ago is absolutely important, as well as literature published 50 years ago, because we have this tremendous opus of literature that gets continually added to, but as it's added to, it is not read. So in some ways, it's like having a DNA methyltransferase run amok, methylating and methylating CpG islands, but then no bromodomain or MBD to read those methylation changes. Therefore, what happens to all those changes? Well, they go unread, undetected, and sometimes the reading of that methylation change will be a necessary alteration of gene expression to survive, or could lead down pathways that are pathogenic, right? So you see, I like that metaphor about not being able to read the scientific literature, just like a bromodomain might, if it's deficient, for example, wouldn't be able to read a rampant DNA methyltransferase activity gone mutated on a spree of methylation of CpG islands. And these things can occur in high stress in cells. We've discovered that oxidative stress is a key feature there. There's a good organic chemical reason for that, but I'm not going to get into it. I just thought about it, but I'm not going to get into it because I'm trying to give you this Coda lecture, and we're still getting into a lot of detail. All right. Now, inhibition of the 1011 translocase mediated DNA methylation process is by mutant isocitrate dehydrogenase, either 1 or 2, making this 2-HG. Okay. So there's isocitrate. From the TCA cycle, got alpha ketoglutarate, isocitrate the alpha ketoglutarate, right? This is the IDH enzyme. This is the dehydrogenase enzyme. This is the TCA cycle, right? Of course. All right, now check this out. When isocitrate dehydrogenase forms alpha ketoglutarate, because of these unusual IDHs, 1, 2, the alpha ketoglutarate then is reacted on with the IDH 1, 2. Okay. Now you make this 2-HG. Now this is the unusual product. Now when you make 2-HG, before we make it, let's take a look at what alpha ketoglutarate is doing normally. When you don't get this double reaction from isocitrate to alpha ketoglutarate to 2-HG. Okay. 2-hydroxyglutarate. That's a normal process of the activity of these two set of enzymes. Now, this one in purple is the one that was picked up being essentially altered, right, in these tumor cell lines. Well, it's got the asterisk by it, it's in a different color. So normally, alpha ketoglutarate is associated with histone demethylases. And those histone demethylases would do what? They would remove methyl groups, right? So if the histone demethylase isn't functioning, right? So if it isn't functioning, you don't get the removal of the methyl group. And the way it doesn't function is if you block it with 2-HG. Yeah. So that's what happens. So 2-hydroxyglutarate competitively inhibits the function of alpha-ketoglutarate-dependent enzymes such as TET2 and histone demethylases like these various ones, JMJD3, FBX11, and KDM2A. So the IDH12 asterisk mutant I just mentioned to you, and that's a mutant IDH1 and mutant IDH2, get two different isoforms. And you also have a couple of other enzymes involved here that are linked to the methylated cytosine. I want to show you where that is, the KME. Where is that enzyme at? Here, oh, they're just showing you where the lysine is, right? So that's the methylation pattern from the original DNA methylation reaction, the methyltransferase. So that's the KME, and we know what 5-methylcytosine and we know what the TCA cycle is. Okay. Now, normally, what you would have with alpha KG, it would be functioning on the TET enzyme, and ultimately, because it goes through those oxidations of the methyl group, remember the oxidations, to the hydroxyl, to the formyl, to the carboxylic acid, just an oxidation of the C1 carbon. And that's what TET2 keeps on cooking away, and you'll get back with a native functional cytosine. But when you inhibit TET2, because you've used this 2-hydroxyglutarate, right? This will inhibit the alpha Kg mediated activation of TET2, just as it will inhibit the histone demethylase, right? So you won't get demethylation here because it's inhibiting the histone demethylase because of the production of 2-HG. So it's not 2-HG, it's actually blocking the activity. It's doing two things. 2-HG blocks the activity of alpha-ketoglutarate, but you're also getting the conversion of the alpha-ketoglutarate. You see how that works? Right? So it's a double negative. Again, off in that Cancer Science paper. All right. Now, move back up to more current times, Frontier in Immunology. It's a paper published in February of 2020. So it's getting on to 2 years almost, right? And you know that can be a long time in publication, right? So T-Reg transcription factor, FOXP3 genetic mutations are known to lead to autoimmune diseases. All right. FOXP3 positive, CD127, CD25 high, okay, is the molecular signature for a T-Reg. So 3 positive, CD127 minus, CD25 high or bright. That's the molecular signature for a T-Reg cell. Remember what T-Reg cells do? They suppress the Th1 and to some extent also the CD8 positive T cell lineage, those canonical pro-inflammatory pathways. That's what T-Regs do. Remember, we talked about these. So FOXP3 transcription, as it turns out, requires a functional and a persistent T cell receptor stimulation. Analysis of FOXP3 positive and FOXP3 minus T cells, because remember, you have to make that transcription factor for the T cell to become a T-Reg cell, right? So a FOXP3 negative T cell isn't going to be able to flare into a T-Reg cell because it doesn't have the right transcription factor that would lead to all the downstream transcription pattern, which is the T regulatory cell. So analysis of those two cell lineages suggests that many T-Reg specific genes don't somehow even require FOXP3. I mentioned this in the lecture a few days ago. So now we have a suite of genes in what could be called loosely T-Reg genes, although they're not expressing FOXP3. They have some of the functions of a T regulatory cell, but not all the processing and maybe growth factor stimulation and alteration of the T helper cells and CD8 lineages, because they don't express the other suite of proteins that were otherwise produced in non-FOXP3 transcriptional activation motif, or maybe not even any nascent transcription factor being expressed, but maybe just housekeeping transcription factors for those cells. So that means ultimately, and we ended last time on this, and we're going to end on it today too, that FOXP3 could be an either or protein in T regulatory signature activity. Okay, and by that, what do I mean? Remember, I told you that either or comes from a book published from a 19th century existential philosopher named Kierkegaard, Soren Kierkegaard. And the book, and what the book's about, I recommend reading it. It's a great book. It's not about, unfortunately, it's not about the immune system, at least not, wasn't intended to be, and it doesn't have any biochemistry in it that I can see, although I do use it, as I am right here, as a metaphor for biochemical and pathobiochemical processes. But the either or signature, I've decided, is a really interesting and unique one. Not even sure that Kierkegaard meant it this way, but this is how I interpret it. When you say either, and we mentioned this last time in the video lecture, you can say either polar, nonpolar. Now, you don't need a copula between those two. You can say polar, comma, nonpolar, right? Now, those are opposites, correct? Of course they are. Or you can say maybe anionic, cationic, right? So there's a polarity, but one is negatively charged and the other is positively charged. Or, that's the either, you can have a hydrophobic response. So either the ionic properties of either cationic or anionic, or hydrophobic interactions. And that's what I see here now, past the one metaphor, one level now further down. Talk about cells, not about inorganic chemistry, right? Polarity and hydrophobicity and things like that. Let's just talk now about these T-Regs. The T-Reg could be a T-Reg with very potent suppressor activity, a T-Reg with almost a TH cell-like of activity, which I will mention to you. It's an alteration of gene expression of that FOXP3. So you have a T-Reg cell that otherwise looks like a T-Reg cell and lots of others of its molecular signature. But because it's not expressing FOXP3 in adequate amounts, it doesn't act as a suppressor of inflammation. It actually enhances that. In fact, those kinds of T-Reg cells are actually at some level interconvertible with Th17 cells, which can be very potent in inflammatory responses, particularly in virally challenged cells. Or those are the eithers, right? Or the T regulatory cell can be doing something totally different. And that different is immunomodulation. So rather than suppressing or activating inflammation, that's the either, it's the comma between the two, the polarity, for example, or the T-Reg signature lacking a sufficient amount of FOXP3 activity could be involved in a completely different aspect of the immune response, a modulatory aspect, which basically means altering the infection court or the stress court of the system, or maybe even just acting in a way to alter the expression of genes in other T cell lineages. Right? Modulating. Okay. So I'm going to stop with that because this basically shows you, we can take a look at this real quickly. The paper published in Frontiers in Immunology, February 2020. Okay, so going on almost 2 years ago, right? Remember we talked about these these non-coding regions, right? These regions of DNA that are part of the entire locus of FOXP3. So you have a CNS0, the promoter itself for the gene FOXP3. Then CNS is 1, 2, and 3, right? These are non-coding sequences, right? Non-coding constitutive sequences. Now, we left you last time with a description of this. Now we're showing you the detail. What the heck, I'll go through it. Because the reason I did all of this epigenetics today was to give you an example. So I've got to give you the example, right? I don't want to leave you without the example. So here you go. All right, move it over some more. Finally, good. All right. Again, these are constitutive non-coding sequences, all part of the gene for FOXP3, right? So you have so you can understand this being blocked by histones, right? Because you know the DNA and chromatin is held together in nucleosomes with histones. And you have histone acetyltransferases. And when they're active, they're going to acetylate those histones on those lysine residues. And you're going to have active chromatin. With that, you're going to have these various proteins, this SALB1 associated with this first non-coding sequence. That's before the promoter. Then remember, we were talking all about the NFAT and CREB. That's a cyclic AMP response element binding protein. P65, straight out of central casting for cell cycle, right? CREL is also there. SMAD, another one. We talked about all these. The MML, Mili4. There's a CPG island right there. If you have miR15A, that's a microRNA, it can lead to FOXP3 expression. And what is it doing here? It's altering DNA methylation, demethylation machinery. Now that is RNA, single-stranded RNA, that's normally thought to associate just with modifying translation. Here it's affecting DNA methylation at the promoter region, okay? So FOX3E2 transcription then can be blocked if you have a deacetylase. That's one way of an epigenetic modification. Remember, when you remove the acetate, you have euchromatin, which is chromatin that's more open and exposed, and that chromatin can be retailored and opened up so that an enzyme like RNA polymerase can come in and transcribe that gene, right? Along with single-stranded binding proteins and the unraveling of the gyrases and topoisomerases to make that transcript work functionally, right? Enzymes have slightly different names when you talk about transcription and you talk about replication, but you get what I'm saying. Enzymes which unravel that chromatin and allow for RNA polymerase to come in and to make that transcript. When you deacetylate at that level, what you have is inactive chromatin. That's one way where you get control. First way I talked about is microRNA 15A16, regulating DNA methylation here on that promoter. And we know that if we methylate a cytosine residue, it's not otherwise hydroxylated through that TET process. It's probably going to suppress the expression of that gene. Right? But if you deacetylate, you're going to shut the whole thing down. Now look at the rest of these regions, these non-coding sequences 2 and 3. You have multiple proteins. There's a TET protein on there. We already know what it does. It's going to modify the methylation pattern on those CPG islands. What's it going to do? It's going to make the hydroxy, 4-mil, carboxy, right? Cytosines, 5-methylcytosines. And what's that going to do? It's going to remove that methylation pattern. Right? So that's when TET is found in CNS2 for those CPG islands. This ENO1 is also going to be, if it's bound on that site, like if it was bound here, I forgot to mention this, it'll block transcription right from the promoter. It'll block transcription at the CNS portion of this DNA, right? That protein acting in trans, right? That's a negative transcriptional control. That's what all these are here. Now, if you have active chromatin, you're going to have the demethylases. So the demethylase, again, will be working along with the acetylase to give you fully active chromatin. But if you have methyltransferases, you put methyl groups on this histone portion associated with the CNS for this gene, still all talking about one gene, FOXP3, you're going to repress it. Repress it with methylation, repress it with deacetylation, promote it with acetylation. Control it by microRNA methylation, demethylation patterning, that's at the level of RNA, and then the TET2 protein controlling the oxidation state of the methyl group here. Okay. So you can see here these various microRNAs. They can block these various reading frames. There's miR17 functioning on these three proteins. There's the IRF4, the EOS, and the SATB. Remember, these proteins are also part of the transcriptional machinery. So these proteins, this one RNA, miR17, not miR15A16, but a different microRNA, is going to block expression of all those proteins. So you won't make those proteins, right? So FOXP3 transcriptional complex formation, you're going to have now, right, miR15A, miR210, miR24, miR35, all at the untranslated region, 3' at the end of that open reading frame. And that's going to block translation. Right? But FOXP3 itself, when it trimerizes, the product of this whole transcription and translation ultimately, it trimerizes, it will block at miR31, and the miR31 can't bind, and that will then de-inhibit this translation. So there's multiple processes. This is a translational level for microRNA. That's how the miR16 works. But it's also indirectly controlling the methylation pattern because it's yielding different changes of expression of these proteins, which are necessary to control the methylation pattern. You see, now that's all just for one transcription factor, FOXP3, regulating T-Reg activation to be a suppressor T cell lineage to control inflammation. Now, if I tell you that the aging process in humans leads to lower levels of activatable FOXP3 plus fully functional T-Reg cells, thus not being able to modulate or control the either-or system over the suppression of an inflammatory response, and you get to where you have multiple sites of inflammation and various tissue beds because different cells are going to have a dysregulation there because of the FOXP3 expression, and the T cells being associated with a potential activation because of innate immune cell responses to, let's say, an infection, with the inappropriate amount of T regulatory cell function, you can have a hyperinflammatory response subsequent to all this, if your T-Reg isn't working functionally, and that can lead then to high morbidity and death in certain disease states, all of which can be linked back to aging because of the lack of all this fine-tuned control over the FOXP3 gene, which is the transcription factor for fully active, non-compromised T-regulatory suppressor agency. Okay. So I'm going to stop there. I got into a lot of detail about that TET protein. I didn't go as much detail as I can, I might mind you, because I do have lectures that talk all about the TET protein in leukemia. And there's the why it's called the 1011 and all that, but I didn't do that today, so I held back. But I can see I'm going to have to do more than one other canonical video lecture for my Coda for aging and the neuroimmune responses in association with environmental interactions with the genome, the epigenome, to lead to senescing morbidity and mortality in humans. I can see that I've got a little ways to go there. So I'm guessing now we're going to be looking at not one, but two more video lectures. Okay. So we have a ways to go. But I think that you're beginning to see the pattern I'm trying to create here. And the pattern, of course, is that when we have a active immune response that is not compromised, that is functioning under normal coordination with all the possible interactions that are necessary for it to function in that motif, right? So remember, in order for the T cells to function, they have to have a controlling mechanism for suppression, as well as an immunomodulatory effect to maintain the methylome and the acetylome and various other epigenetic modifications. We didn't talk yet about how those microRNAs are expressed, but you can believe that they're also under a similar control in highly reactive cells such as T lymphocytes. And they are, and we will cover that eventually, if not in a video lecture, certainly in an audio lecture. Okay. So I'm going to leave you with that today. I don't know how much time we spent, but I hopefully have brought more of this discussion into detail. Sorry about the coughing at the beginning. It was just clearing my throat. I'm not ill or anything. Today is that prime day, the 7th day of the 11th month of the year 2021. And hoping you're going to have a good weekend, what's left of it. It's already almost Sunday afternoon here, even with the hour change in good old Northwest. So this is Dr. Dan Guerra from Authentic Biochemistry Studios, wishing you a very pleasant and blessed Sunday afternoon, saying bye for now.